the session. I've started the recording. I don't know who was the bioanal guy, but then or a girl, but suddenly he or she has disappeared. So let me share the link. Okay. So the topic that we are supposed to discuss now is cDNA preparation and its uses in the gene analysis. Okay, and by far the journey looks very good. So we have learned many things in terms of molecular biology, transgenic animals for that matter, and then for, also the last lecture was on production of monoclonal antibodies. So maybe we are not learning it in very detail as that biotech student or means MSc biotech or MSc microbiology students might learn, but this is just to give you an idea about what all techniques and how far the molecular biology has reached. So even if you think of taking up any molecular biology sector jobs after your MSc, you should be prepared for the same. Okay. So the next topic for your information that we are going to do is cDNA preparation and its uses in the gene analysis. Okay. So cDNA preparation क्या रहता है? What are cDNA libraries and how they are different than that of the genomic libraries, and what are their main uses? That is what we are going to see now. So what is cDNA library? So cDNA library is a combination of cloned cDNA, and the cDNA are nothing but complementary DNAs. So what are these complementary DNA? I will tell you as we move on with the lecture. So first we understand the definition of it. So cDNA library is a combination of cloned cDNA. So when we say clone, that means it is going to be in some host, okay? And it is that complementary DNA is going to be in presence with a vector, okay? So when we have a DNA of interest with the vector, we call it as cloned DNA, right? So cDNA library is a combination of cloned cDNA fragments inserted into a collection of host cells which together constitute some portion of the transcriptome of the organism. So what do you mean by transcriptome? Is the transcript product, right? DNA, when it gets converted into the RNA, it will be called as transcriptome. And I hope you recollect your semester 1 lectures in which we have talked about what is transcriptome analysis, protein analysis, genome analysis. In that we have learned about transcriptome in detail. Okay, so so these are the fragments that are inserted in DNA, cloned cDNA fragments that are inserted into a collection of host cells, which together constitute some portion of the transcriptome of an organism. So basically, it is the expression of the mRNA of a eukaryotic gene into a prokaryote. Okay. So that is why we are building. So most of the time, the cDNA libraries will be produced for the eukaryotic cells. I hope you guys understand the difference between eukaryotes and prokaryotes. So eukaryotes examples could be us, humans, animals, plants, etc. And prokaryotes examples could be bacteria and fungi or something like that. Viruses could be. Okay, now. So cDNA is produced from fully transcribed mRNA found in the nucleus and therefore contains only the expressed gene of an organism. So this is a very important point to be noted down over here, is that cDNA is produced from fully transcribed mRNA only. So when we are saying that mature RNA are the one that are getting converted into complementary DNA. That is what they're trying to say. And we all know that mature RNA is produced after the transcription process the splicing will be happening, and the 5' cap will be put on the mRNA, and poly A tail will be applied at the end of 3' at 3' So that is when you will be calling that transcript will as a mature RNA. ठीक है? RNA के बहुत सारे types रहते हैं, tRNA, rRNA, and mRNA is the one which carries the sequence that are going to get translated and get converted into the proteins and enzymes. So why only mRNA? Because it contains only those sequences that are to be translated, and they have only exons, no introns. So only expressing sequence are going to get are going to get converted into cDNA library. Okay, so that is the main significance of it. So why we are constructing cDNA libraries rather than that of the genomic libraries, we will understand this process. As we understand, I will explain you that also why we are preferring cDNA libraries over genomic libraries while making multiple copies of this cDNA clones that we are going to insert into the host. Okay. Uh, so I hope, have you guys understood the definition of cDNA library or cDNA? So basically you have a DNA. DNA is getting converted into mRNA. From that mRNA, we are going to make cDNA. That cDNA we are going to clone into the vector. Means like, sorry, we are going to put it in the vector. We are going to obtain a clone. And then that clone will be again put back into the bacteria so that it can make multiple copies of the same. Are you guys understanding? Yes or no, you have to tell me in the chat box. Okay. Okay. So that is what the definition about cDNA library is. Then moving on, what is cDNA library? Means in continuation. So in eukaryotic cells, the mature mRNA is already spliced, and hence cDNA produce lacks introns and can be radially expressed in the bacterial cells. So what they're trying to tell you is that if I take this and if I show you on a blank screen, just take, let me share a blank screen with you. New share, whiteboard, share. Okay. So when you have genomic library made, okay, genomic library. So I just denote it as slide L over here. And when I have cDNA library, okay, CL. So in genomic library, whatever I'll be saving into the my library it will be having everything. It will be having exon and also the introns. Okay. But the problem with this setup is that I cannot put this, I cannot clone this and put this into the bacteria for making multiple copies of the same. I can probably clone it into a vector, but I cannot put this inside the bacteria because the bacteria cannot incorporate a DNA which has the introns, okay, and they do not have the mechanism where they can undergo the splicing. It is very specific mechanism to eukaryotes only, okay. So that is the reason genomic libraries are of no use while making when you want to put these kind of clones into the bacteria and make them make multiple copies of the same. But in case of cDNA libraries, as I just explained you, we have mRNA, right? mRNA. It's very difficult to write with this. Okay, so we have mRNA, and we all know that mRNA will be having poly A tail, mature RNA, okay, poly A tail, okay, and it will have 5 dash cap. So this is 5 dash, okay, and this is 3 dash, okay. Uh, can I take a smaller one? So this is your mature RNA and it has undergone. So when we say mature RNA, there are two things that are happening with the transcript. So there is a difference between the transcript and the mRNA also, right? So when my DNA will undergo what transcription, I will get a transcript, okay? So this transcript will have exon and it will also have introns, okay. So this is not something I need for making my cDNA libraries, and this is not something which is going to go and get translated and make the proteins for my body requirements, right? So it has to be modified, right? And that modification is taken care by first procedure, that is splicing, alternative splicing, but we'll call it just splicing over here, okay. And another one is addition of this 5 dash cap, okay, and poly A tail at the 3 dash end. So once these are done, so when splicing, what do you mean by splicing? Only exons will be remaining, okay, and all the introns will be gone. And exons are the sequences which code for a protein, right? They are the coding sequences for the proteins. So this mature this mRNA of ours will be having only exons, no introns, and it will have only coding regions. It will have 5 dash poly A tail. It will have 5 dash cap and 3 dash poly A tail. So this is when I'll be calling my RNA will be as mature RNA, which can go further and translate. Okay. So this is the mRNA that I'm going to take to make the cDNA library. So this particular mRNA, I can easily clone into a vector. I can easily combine with the vector, okay, and make a clone of that DNA, and then I can put it back into the bacteria, because since there are no introns, bacteria will consider them as their own DNA, and will allow them to go ahead and do the replication process, right? So that is the most important part, and that is the reason why why there is a need, why there was a need of constructing cDNA libraries over your genomic libraries. Genomic libraries also have another application. So when scientists have done genomic libraries, which includes everything, the exon, introns, etc. But the cDNA libraries are the one which have only the coding regions, which you can easily put into a vector and make a DNA clone, and then you will be putting it back. So I'm not saying that this mRNA will put directly into the vector. This mRNA has to be converted into the DNA. And when we convert this mRNA into the DNA, it will be called as the complementary DNA. What it will be called as? It will be called as the complementary DNA. This complementary DNA then will be inserted into the vector. Okay, so this is where your cDNA will be. Okay, and then that will be inserted back into the bacteria. Very clumsy diagram, but I hope I have made this clear. What exactly do you mean by uh, cDNA library? Okay, means like how exactly we are going to go and understand this procedure. Let me share this slide again. Okay. So in eukaryotes, so since I have explained you this procedure, we can go ahead and read this statement, what they're trying to tell you over here. So in eukaryotic cells, the mature RNA is already spliced, and hence the cDNA produced lacks introns and can be readily expressed into the bacteria. So it is the cDNA that we are going to get after converting that mature RNA into the DNA with the help of reverse transcriptase procedure that I will let you know how exactly that works. So it will be radially expressed into the bacterial cell. Okay, so if you have a DNA which has introns as well, it will be very difficult to be expressed into the bacteria. And that is the reason you convert the mRNA into the complementary DNA with the help of reverse transcription, and then put those back, put those cDNAs into the vectors and put those vectors into the bacteria so that they can multiply. Okay. So while information in cDNA library is a powerful and useful tool, since gene products are easily identified, the libraries lacks information about enhancers, introns, and other regulatory elements found in a genomic DNA library. So, but since these ones have only exon data, they will be lacking all other part. Okay, they will be lacking enhancers data, introns data, मीन्स जो कुछ भी एक्सॉन छोड़ के और कुछ भी रहेगा जो डीएनए में, वो सारा इंफॉर्मेशन दे विल बी लैकिंग, दिस cDNA libraries will be lacking. That is what they are trying to tell you over here. Am I clear till here? Especially the chemistry people, you have to let me know, because this is something new and I don't know whether microbiology students have ever talked up, means like have any good idea about it. Do you guys have any idea about it? Sheetal, Pratibha, Darshana, have you ever learnt about cDNA libraries in your biotech or microbiology syllabus? Okay, okay, okay. So yeah, but I'm sure the chemistry people have not done it ever. Okay. Still, you guys are here and learning more about this. So I hope I'm giving you more understanding in this part, particularly cDNA libraries, and making it clear. So are we clear till this part? What exactly do you mean by cDNA libraries? Okay. So we will sum up the procedure again as we move further. Okay. So intron, exon in eukaryotes. So we see over here, this is uh, 3 dash N, this is the 5 dash N, and then we have a DNA, it has exons, it has introns, it has promoter, that is starting sequence, this is the stop code, starting sequence, termination sequence, and it undergoes the transcription, and then transcription then makes the mRNA. Okay, then mRNA undergoes the processing to make it mature. Okay, to make it translatable. Okay, though after this mRNA undergoes processing, you can see over here the cap will be added at the 5 dash end. This is the 5 dash end. Now they have changed the direction of the arrow, and the poly A tail will be added, and this intron sequences will be deleted. Okay, that is what is called as splicing. And then we have mature mRNA, which will go to the cytoplasm, and this whole process of splicing of mRNA takes place inside the nucleus. And once we have mature mRNA, it goes to the cytoplasm. I hope I have made it very clear over here, and I have made it clear in our first and second semester as well. So this is how the diagrammatic representation of formation of mature RNA they have shown over here. Okay, just a revision over here. Okay, so cDNA library preparation. So cDNA is created from a mature mRNA from a eukaryotic cells with the use of an enzyme known as reverse transcriptase. Okay, so now very simple fundamental is to be applied over here, or the revision I would do it for you people, is that when you convert DNA to RNA, you will be using enzyme called transcriptase, okay, or RNA polymerase, okay, because you are going to form the RNA from the DNA. But now you want to reverse the procedure. So you will be simply using reverse transcriptase. So transcriptase is an enzyme which is required for conversion of DNA to RNA, whereas reverse transcriptase over here is an enzyme which is required for conversion of RNA to DNA. Okay, so that is the main important part in understanding of making of cDNA libraries. So cDNA is created from a mature mRNA from a eukaryotic cell with the use of an enzyme known as reverse transcriptase. And in eukaryotes, a poly A tail consisting of a long sequence of the adenine nucleotides distinguishes mRNA from the tRNA and rRNA. So that is also most important part. So mRNA, we know there are types of RNAs, and you want to distinguish between the tRNA, RNA, rRNA, and mRNA. So we know that mRNA are the one who are having this poly A tail sequences. Very when I'm saying poly A, that means यहाँ पे A ऐसा sequences रहेंगे, right? That is what makes mRNA distinguishable than that from than that of your tRNA and rRNA. So in eukaryotes, a poly A tail distinguishes mRNA from tRNA and RNA and can therefore used as a prime site for reverse transcription. So that site can be used as the site where the primer will come and bind, and we can achieve the reverse transcription. Okay, so now see, we all know that mRNA will be single stranded, yes or no? Okay, now from this single strand you want to make what? Double strand, right? DNAs are double stranded, right? So for that you will be needing a primer. What do you mean by primer? I have told you guys what do you mean by a primer. Just tell me in the chat box. I don't want answer from biotech and microbiology student. I want answer from chemistry people. What do you mean by primer? Don't Google it. Don't fool yourself. What do you mean by primer? Yes, Akash, Shraddha, Samruddhi, Razina, you guys have to tell me what do you mean by primer? Tanuja, no. Single-stranded nucleic acid, no. No, that is not Supriya. What do you mean by primer? Naina, you can tell us.
Ma'am, it initiates the replication.
Yes,
So
it initiates
it's
the
the first
replication.
part which is needed.
Yes, yes, yes. It is basically, if I tell you, the primers are nothing but a small sequences around 20 nucleotides, which is required to process the further thing. That is in the sense to do the further transcription or translation, whatever you guys, transcription mainly, या फिर replication. Okay, so it is the 10 to 20 nucleotide base pair sequence. Okay, since we see this part, you have to understand, you have to know what do you mean by primer, you have to know what do you mean by when I say nucleotide sequence. तो तुम्हें पता रहना चाहिए कि nucleotide क्या है exactly, ठीक है? Because then only you these things will make sense to you. Otherwise, it will be very difficult. Chemistry people, you have to go back and learn you from your 12th standard books if you still are confused with nucleotides and sequences and stuff like that. It's okay to be confused. There is nothing wrong in it, but you have to have a clear idea about it. Understanding? Because this will create problem in your understanding. And what is my thinking is that even if it's not of your interest, but it is there in your syllabus for some purpose, so you should understand. Okay, and we are into bioanalytical sciences, which is an interdisciplinary. That means when we say interdisciplinary, that means it is going to give you a knowledge of each and every part of the industry area that are in work, means that are in progress right now. Okay, so how we talked about, and maybe the analytical chemistry and whatever we did, HPLC was a very difficult task for the people who have come from biotechnology or microbiology background, but still they could do it. I understand the molecular biology to concepts are very difficult to understand, but you see, watch some videos, and then you have to have that background to be very clear. Then only it will make sense to you. Okay, so we will go back to our topic. Okay, so it's not very difficult thing to understand, but just that it involves lot many definitions and lot many tiny things to be remembered all the time. Okay, so so uh, can be therefore used for primer site. So we were talking about the poly A tail. Okay, so now we know that mature RNA is having what? Poly A tail, right? It has poly A tail. A. तो मुझे अभी ये सिंगल स्ट्रैंड को क्या बनाना है, डबल स्ट्रैंड बनाना है, राइट. आई वांट टू डू द रिवर्स ऑफ ट्रांसक्रिप्शन. ओके, reverse transcription. That means in transcription, I'm converting a DNA into RNA, and now here I will be converting what? RNA into what? DNA. ठीक है, and this mature RNA will be selected for that process. You can do it only with mature RNA. You will not be doing it for tRNA or rRNA. Okay? What is the advantage of selecting mRNA is that first of all, it has only coding sequences. Okay? It has only exons. It doesn't have introns. It doesn't have enhancer, promoters, etc. Okay? Then second advantage that it is very much secured. It has undergone the splicing thing, and it also has a poly A tail. Okay? Now I was talking about the primer. So the primer that I have to design, which will come over here and process this particular strand, right? See, when I'm saying reverse transcription, what is going to happen? यहाँ पे जितने भी सारे न्यूक्लियोटाइड रहेंगे, उसको कॉम्प्लीमेंट्री न्यूक्लियोटाइड्स यहाँ पे फॉर्म होंगे. Yes or no? ठीक है. So when I'm going to design a primer, it will be a very easy task for me because I just have to design a primer with a T, 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 and T. So this TT primer, starting primer, will immediately come and bind to the poly A tail, and the transcription, reverse transcription will be started in this direction, right? So as my reverse transcriptase enzyme will be moving over here, it will be recruiting the nucleotides which are complementary to the nucleotides which are present on the mRNA strand. Okay? So that is how the double-stranded DNA will be formed. Okay? So let me see if they have explained anything further in this aspect. So yes, we can read out over here and then we can talk. Let me clear this. So I hope I have made this clear how exactly the primer will be attaching and will help into the reverse transcription process. Next is mRNA extraction. So firstly, the mRNA is obtained and purified from the rest of the RNAs. That means you will be sorting only mature mRNA and you will be excluding tRNA and rRNA. Several methods exist for purifying RNA, such as triazole extraction, column purification, etc. Column purification is done by using oligomeric DT nucleotide coated resins where only mRNA having poly A tail will be binding. So you can see over here. See, this is the diagram where it has oligo DT matrix, and only so this is the mixture of all the RNAs you have, mRNA, tRNA, rRNA. So only things that will be binding to the oligo DT matrix will be your mRNA since it has poly A tail over here. Okay? So we go back to that particular reading. So the rest of the RNAs are eluted out, and the mRNA is eluted by using eluting buffer and some heat to separate the mRNA strands from oligo DT. So this is how the mRNA extraction is done from the mixture of RNAs that you will be getting from the nucleus. So this is where you can separate the mixture of RNAs only specifically for your mRNAs. Okay, and that is what they have shown in the diagram. Then purification can be performed by binding mRNA on a solid matrix to which short string of thymidylate residues are attached, that is oligo-DTX matrix, and the mRNAs are removed again by washing in a low salt buffer. So this is what they have shown over here. So you have mixtures of RNA, you will be having this oligo-DT matrix to which only the mRNAs will go and bind, because you see the complementary T's are here and we have complementary A's over here. Then mixture of so mix under the hybridization condition. So only you see mRNAs are getting bind over. Wash away RNA and tRNA, then elute the column in a low soil buffer. So elution. So this is the very what you say. It is high grade technique that we can use, where we are using a solid matrix to extract the mRNA from the mixture of RNAs. You have anything to ask, Akash? You can ask. So this is how we'll be getting the purified mRNA, by using a solid matrix which is coated with the oligo DT. Okay, we will be stopping over here the mRNA extraction part. So this is for making cDNA library. The first thing that we have to do is mRNA extraction. Okay, then after mRNA extraction, we'll be doing the further procedure. Are we clear till here? You have anything to ask, Akash? You can type it in the chat box probably. Okay. Okay. So I hope this diagram is very clear. Is it clear? Yes, Supriya. Okay. So what we have done by far is we have understood what do you mean by cDNA library, why you want to select cDNA library over genomic libraries, and what exactly you will be doing. The first step to cDNA library production will be your mRNA extraction. That is what we have seen. So further part to it we'll be discussing in the next lecture. So tomorrow we are meeting through the Google Classroom, Google Meet, I mean to say. So yeah, if there is little bit of a hassle, just don't panic. I'll be sharing the link and everything with you, mine and Kalyani ma'am's. The timetable is going to be the same. Maybe 5-10 minutes extra on the classes we can exceed. Okay. So we'll keep in mind that you must have a little bit of a break between two sessions. Okay. So that will be taken care of. So I'm signing off for now. If you have any doubts, please come up with your doubts tomorrow and we can discuss the same. Okay. I'll see you all tomorrow.